Hello guys, welcome to my YouTube channel, and in today's video we are going to talk about glycolysis. So this is going to be a very interesting video, but before we go further, you need to show me support. Make sure you subscribe to my YouTube channel, make sure you like, and make sure you comment. Guys, I really appreciate that because that's the only way you're going to support my work. Alright guys, let us get into it. So first of all, we need to understand what glycolysis is. So what is glycolysis? So glycolysis can be simply defined as the breakdown of glucose in order to produce a molecule that we call ATP. So we're breaking down glucose in order to produce ATP. What else are we producing in glycolysis? Or what's the other purpose of glycolysis? So the other reason we're having glycolysis is to produce pyruvate. So we are producing ATP and pyruvate as well as NADH. So glycolysis can be defined as the breakdown of glucose in order to produce ATP, pyruvate, and NADH. Now, when does it take place? When does glycolysis take place? So glycolysis takes place all the time, but it increases. The rate of glycolysis increases during well-fed state. So what is well-fed state? So well-fed state, that is after you're from eating a meal rich in carbohydrates. So after eating a meal rich in carbohydrates, what happens that the carbohydrates, they go in the GIT where they're broken down into glucose, and that glucose is absorbed into blood. So when glucose enters circulation, it goes to the pancreas. And you know what? In the pancreas, we have got cells that we call beta cells. So why are these cells important? The beta cells, these are cells that produce insulin. So when there's high level of glucose in the blood, it stimulates the beta cells to produce insulin. And now, when we produce insulin, it goes into circulation as well. So now in the blood, we have got insulin, high level of insulin, and high level of glucose. Now, why are we producing insulin? So insulin, it facilitates glucose uptake in cells. But now, what are those cells we're talking about? Where does insulin go? So we have got cells that are sensitive to insulin. So the main cells that are sensitive to insulin are the liver cells. You should take note of that. So the main cells that are sensitive to insulin are the liver cells and the muscle cells as well as the fat cells. So the liver cells, muscle cells, and the fat cells. These are the main cells that are sensitive to insulin. So insulin, it can go to the liver, it can go to the muscle cells, and it can go to the fat cells. Now, what is it going to do to the muscle cells and the fat cells? That's a good question. So when it goes to the muscle cells and the fat cells, in the muscle cells and fat cells, we have got what we call GLUT4 transporters. Now, these GLUT4 transporters, they normally reside inside the cell. So they are found in vesicles inside the cytoplasm. So you can see that is a vesicle containing the GLUT4 transporters. So these are simply channel proteins. So I've got GLUT4 transporters in the cytoplasm. Now, these GLUT4 transporters, they are responsible for uptake of glucose into the cell. Now, they are found in the cytoplasm. So what happens that insulin, when it comes, it's going to bind the insulin receptors on the cell membrane, and then it's going to tell these guys, the GLUT4 transporters, to come and bind to the cell membrane. So when the GLUT4 transporters, they come, they're going to bind to the cell membrane, and glucose is going to enter. So that is the role of insulin. So you should know that we have got a lot of GLUT transporters. There are a lot. Now, we have got from GLUT1 up to GLUT14. So there are a lot. And each type and each isoform of the GLUT transporters is found in specific tissues. So for example, when you talk about the GLUT1 transporters, these are found in red blood cells. When you talk about the GLUT2 transporters, they are found in the beta cells as well as the liver cells. When you talk about the GLUT4 transporters, they are specifically for the muscle cells and fat cells. That's why we are going to find the GLUT4 transporters. And then you guys, what you should know is that out of all these 14 GLUT transporters, only the GLUT4 transporter is insulin sensitive. So only the GLUT4 transporters are controlled by insulin. The rest are not controlled by insulin. So that should tell you that the rest of the GLUT transporters, they are already found in the cell membrane. They are embedded in the cell membrane, except the GLUT4 transporter, which is normally found in the cytoplasm or in the cytosol. Okay, so we have seen the role of insulin. So insulin, it is produced during well-fed state or when there's high level of glucose in the blood, and then it stimulates the uptake of glucose into the cells, especially the muscle cells and the fat cells, because we know that these, they are the ones that have got GLUT4 transporters, which are insulin sensitive. Now, what is happening when insulin enters the cell? That's another question. What is going to happen? So when insulin enters the cell, something interesting is going to happen. You guys should know that the glucose transporters, these are bidirectional. So it means that glucose can enter and leave. So they are bidirectional. So what are we going to do to trap this glucose inside the cell? We are going to add a phosphate group. So there is an enzyme that is called hexokinase. So hexokinase, what is it going to do? It's going to add a phosphate group to glucose so that we prevent it from escaping to the outside of the cell. So we are going to add a phosphate group. But now, where are we getting that phosphate group? Where is it coming from? So here, we are going to break down what? ATP. So we are breaking down ATP. We are going to break down ATP. And form what? And form ADP. So we are going to grab a phosphate group from ATP and add it at carbon number 6 in a glucose molecule so that we form glucose 6-phosphate. Now, what enzyme is doing that? There's an enzyme here that we call hexokinase. Now, in some texts or some textbooks, you're going to find that they put another enzyme which is called glucokinase. Now, what is the difference? What is the difference between hexokinase and the glucokinase? So the difference between these two enzymes is here. When you look at hexokinase, this is an enzyme that is found in all cells except the liver and the beta cells. So you're going to find it in all cells except the liver and the beta cells. That is hexokinase enzyme. What's the other thing? Hexokinase is not insulin sensitive. Now let us compare it to glucokinase. So hexokinase is found in all cells except liver and beta cells. Glucokinase is found only in liver and beta cells. Have you seen the difference? Hexokinase is not insulin sensitive, so it doesn't get controlled, or it is not controlled by insulin. Hexokinase is not controlled by insulin. But glucokinase is insulin sensitive. What do we mean? Glucokinase is controlled by insulin. So these two enzymes, they are isoforms of each other. So you should know that hexokinase, it acts on exosugars. So it can act on any exosugar, any sugar that is having 6 carbons. Glucokinase, it only acts on glucose. That's why the name glucokinase. So when you're looking at enzymes, make sure you pay attention to the specific prefixes as well as suffixes. Okay, so we've seen the difference. So we know that glucokinase, it is only for the liver and beta cells. Hexokinase, it is for all these other cells. So here we have got hexokinase, which is not insulin sensitive. So this is not insulin sensitive. So it's going to add a phosphate group to glucose, so we form glucose 6-phosphate. Now, from glucose 6-phosphate, there's another enzyme that we call phosphoglucose isomerase. This enzyme is just going to rearrange atoms so that we form an isomer of glucose 6-phosphate. What do we call that isomer? We call it fructose 6-phosphate. So here there is fructose 6-phosphate. Now, fructose 6-phosphate can be further converted to fructose 1,6 bis-phosphate by a molecule called phosphofructokinase 1. So this is an enzyme that can convert fructose 6-phosphate to fructose 1,6 bis-phosphate. Now, this enzyme is well regulated. What regulates this enzyme? That's a question that we need to ask ourselves. So, insulin is one of the regulators. So you can look at the arrow there. You can look at the arrow there. You can see what is happening. That is insulin sending signal here. Now, what is happening? How is phosphofructokinase 1 or PFK1 regulated? Insulin, it activates phosphoprotein phosphatase enzyme. So when phosphoprotein phosphatase enzyme is activated, we have got another enzyme here that we call phosphofructokinase 2. You guys should see the difference. This is phosphofructokinase 2 and this is phosphofructokinase 1. So we have got an enzyme that is called phosphofructokinase 2. So this enzyme normally, it has got those phosphate groups attached to it. And when it has those phosphate groups attached to it, it means that it is inhibited. It cannot work. So PFK2 or phosphofructokinase 2, it has the phosphates attached to it. And that simply shows or telling us that this enzyme is inactive. It's not working. Now, what happens is that when insulin comes bind to the insulin receptor, it activates this molecule that we are calling phosphoprotein phosphatase. So a phosphatase enzyme, it is an enzyme that is removing the phosphate. So this phosphoprotein phosphatase enzyme is going to remove the phosphates from phosphofructokinase 2 or PFK2. Or in other words, this enzyme is going to dephosphorylate PFK2. And when we dephosphorylate PFK2, PFK2 becomes active. Now, what does it do when it becomes active? Why even having this enzyme? PFK2 is very important because it can convert some of the fructose 6 phosphate to a molecule that we call fructose 2,6 bisphosphate. You guys don't confuse it with fructose 1,6 bisphosphate. These are two different molecules. So, from fructose 6 phosphate, we can either form fructose 1,6 bisphosphate using PFK1, or we can form fructose 2,6 bisphosphate using PFK2. So let me just do a recap. Insulin will bind to the insulin receptor, it will send signals, and it will stimulate this enzyme that we call phosphoprotein phosphatase, which is going to dephosphorylate phosphofructokinase 2 so that this enzyme becomes active and it can convert fructose 6 phosphate to fructose 2,6 bisphosphate. Now, why are we forming fructose 2,6 bisphosphate? So, fructose 2,6 bisphosphate, this molecule, it is an allosteric activator. It is an allosteric activator. When we mean allosteric activator, it means that it's going to bind to the allosteric site of phosphofructokinase 1 and activate this enzyme. When this enzyme is activated, what do you think will happen? Fructose 6 phosphate will be converted to fructose 1,6 bisphosphate. Okay, so normally PFK1, it is sleeping. Normally it is not working. It is inactive. So for this enzyme to be activated, it requires fructose 2,6 bisphosphate to bind to it. When fructose 2,6 bisphosphate binds to PFK1, PFK1 become active and it starts converting fructose 6 phosphate to fructose 1,6 bisphosphate. So that step, it is a regulatory step, and what regulates it, we've seen the activator is the fructose 2,6 bisphosphate. That is the activator. Now, we are saying that this enzyme is converting fructose 6 phosphate to fructose 1,6 bisphosphate. You know what is happening here? We're simply adding a phosphate group at carbon number 1. Now, where are we getting that phosphate group? So we are getting it from ATP. So even there, we are using ATP. We're using ATP and producing ADP. Okay? So we are adding a phosphate group to fructose 6 phosphate so that we form fructose 1,6 bisphosphate. That is what is happening there. Now, we've talked about something that activates this enzyme, the PFK1. We've said that it is activated by fructose 2,6 bisphosphate. Now, what inhibits this enzyme? So the inhibitor of this enzyme are the products of glycolysis and the TCA intermediate. So the product of glycolysis, the main product is what? It's ATP. So ATP is going to inhibit PFK1. Okay, so you guys should know that ATP will inhibit PFK1. What else? Citrate, which is a TCA intermediate. Citrate will also inhibit that enzyme. So citrate and ATP are the inhibitors of PFK1 enzyme. The activator of PFK1 enzyme, it is fructose 2,6 bisphosphate. It's the same as insulin. So in some books you are going to see they have said that insulin is the activator of PFK1. They are simply meaning that insulin, it activates PFK1 using fructose 2,6 bisphosphate. So if you forget fructose 2,6 bisphosphate, you can mention insulin as the activator of this enzyme, because insulin, it activates this enzyme through this molecule. Okay, I think I've explained on that. Now, what is the other inhibitor of this enzyme? The other inhibitor of this enzyme, we can say glucagon. But how does it do it? So glucagon, it is going to activate PKA, protein kinase A, through the CAMP pathway, of the G protein coupled receptors. So glucagon is going to activate the protein kinase A enzyme through the G protein coupled receptors. So if you remember the CAMP pathway, we activate protein kinase A. So when you activate protein kinase A, what does it do? Protein kinase A, it's going to add phosphate groups. To which enzyme? To phosphofructokinase 2. So PFK2, when we add phosphate groups, it's going to be inhibited. Because I said that this enzyme, when it is dephosphorylated, that's when it becomes active. Now, pKa will add phosphate groups to PFK2 and then it becomes inhibited. When it becomes inhibited, what do you think will happen? You cannot produce fructose 2,6 bisphosphate. You can't produce that. And if you can't produce that, are you going to have an activator of PFK1? You're not going to have activators of PFK1. So if you inhibit this enzyme, PFK2, you cannot produce fructose 2,6 bisphosphate. And without fructose 2,6 bisphosphate, you can't activate PFK1. And without PFK1 being activated, you can't convert fructose 6-phosphate to fructose 1,6 bisphosphate. As a result, you're going to inhibit what? The glycolysis pathway. So glucagon is an inhibitor of glycolysis. Insulin, it is an activator of glycolysis. Okay? What else inhibits glycolysis? Citrate and ATP. Okay, so we can proceed. So after forming fructose 1,6 bisphosphate, where do we go to next? So we are going to form what we call dihydroxyacetone 3-phosphate and glycerolaldehyde 3-phosphate. So these are 3 carbon molecules. So dihydroxyacetone 3-phosphate, it is a 3 carbon molecule. And it is simply an isomer of glycerolaldehyde 3-phosphate, which is another 3 carbon molecule. Now, the enzyme that is here is called aldolase. You guys usually should know that dihydroxyacetone, we don't have enzymes that can break down dihydroxyacetone in glycolysis. But we do have enzymes that can break down glycerolaldehyde 3-phosphate. So enzymes are there that can act on glycerolaldehyde 3-phosphate, but not on dihydroxyacetone 3-phosphate. So what will happen? So when you produce these 2 molecules, this glycerolaldehyde 3-phosphate, it can be used in glycolysis. There are enzymes that can break down that molecule. But we don't have enzymes that can break down dihydroxyacetone 3-phosphate. So what will happen is that this dihydroxyacetone 3-phosphate will have no other choice apart from being converted to glycerolhaldehyde 3-phosphate. But glycerolhaldehyde 3-phosphate can also be converted back to that. So there is an interconvention here. So here I've got an enzyme that we call triose isomerase. So triose phosphate isomerase, that is an enzyme that is going to convert dihydroxyacetone 3-phosphate to glycerolhaldehyde 3-phosphate. Now, when we make glycerolhaldehyde 3-phosphate, what is going to happen? It's going to be converted to a molecule called 1,3-bisphosphoglycerate. So this molecule is simply telling us that we have got a phosphate group at carbon number 1 and carbon number 3, as you can see there. Okay? So everything else here, we are going to produce 2 of them. What do we mean? Remember, we have got glycerolhaldehyde 3-phosphate being produced here and dihydroxyacetone 3-phosphate being produced here. So this glycerolhaldehyde 3-phosphate will enter glycolysis and produce a 1,3 bisphosphoglycerate, 3 phosphoglycerate, 1,2 phosphoglycerate, 1 phosphoenolpyruvate, and 1 pyruvate. Have you seen? That's what's going to happen. So from 1 glycerolhaldehyde 3-phosphate, we are going to produce 1 of each of these molecules. Now, after we produce that, this molecule, dihydroxyacetone 3-phosphate, will also be converted to glycerolhaldehyde 3-phosphate. And we are also going to produce another 1 molecule of each of these molecules here. So how many are we producing at the end of the day? So from this, we're producing 1 of each of these, and then from that, we're producing 1 of each of these. So we are producing 2. Okay? So from 1 glucose molecule, we are producing 2 of each of these. Okay, so for example, 1,3 bisphosphoglycerate, we are producing 2. There is that one and that one. 3 phosphoglycerate, we are producing 2 from 1 glucose molecule. There is that one and there is that one. 2 phosphoglycerate, we are producing 2 from 1 glucose molecule. There is that one and that one. Phosphoenolpyruvate, we are producing 2 from 1 glucose molecule. There is that one and that one. Pyruvate, we are producing 2 molecules of pyruvate. There is that one and that one. Have you seen? So everything is doubled here because of the reason that I've stated. Now, you guys want to know that from glycerolhaldehyde 3-phosphate to 1,3 bisphosphoglycerate, there is an enzyme that is called glycerolhaldehyde 3-phosphate dehydrogenase. So this enzyme, what is it doing? So this enzyme is simply removing a hydrogen ion. So it's going to remove a hydrogen ion. And what are we going to make? We are going to make a molecule that we call NADH. So NADH is going to be made there, okay, from NAD positive. So we're making NADH from NAD positive. So what this enzyme dehydrogenase is doing is simply removing a hydrogen ion or an hydride ion from glycerolhydride 3-phosphate and add it to NAD positive to make NADH. So whenever you see an enzyme ending with dehydrogenase, just know that it is involved in the transfer of electrons, or it is involved in the removal or addition of an hydride ion. Okay, that is what is happening there. So we are forming NADH. And remember I said everything down here is doubled. So how many NADH we form from one molecule of glucose? We form 2 NADH. That's what you should know, guys. We form 2 NADH. Now, when we make 1,3 bisphosphoglycerate, what happens to that? So if it is in the red blood cells, 1,3 bisphosphoglycerate, it can have two ways. It can either proceed in glycolysis, or it can go to what we call Rapaport Leubering Pathway. Rapaport Leubering Pathway, or shunt. So if it is in the red blood cells, we can take that 1,3 bisphosphoglycerate into this pathway called Rapaport Leubering Pathway. What is Rapaport Leubering Pathway or Rapaport Leubering cycle? What is it? So this is a shunt of what? Of 1,3 bisphosphoglycerate. So we can get 1,3 bisphosphoglycerate so that we produce what we call 2,3 biphosphoglycerate. So what is happening here that there is an enzyme that is called 2,3 biphosphoglycerate mutase. So what is this enzyme doing? A mutase enzyme, it is simply shifting the carbon. So a mutase enzyme, it is simply shifting a phosphate group. From which carbon? From carbon number 1 to carbon number 2. Okay, so this enzyme is simply shifting a phosphate group from carbon number 1 to carbon number 2. So mutase enzymes are enzymes that are shifting a phosphate group from one carbon to the other, from one position to another. 2,3 biphosphoglycerate mutase enzyme is going to form what we call 2,3 biphosphoglycerate molecule. Now, what is the purpose of this molecule? So this molecule, it is important in red blood cells. And you guys want to know that Rapaport Leubering cycle, it only takes place in the red blood cells for the production of what? Of 2,3 biphosphoglycerate. Now, why are we producing 2,3 biphosphoglycerate in red blood cells? This one, it reduces the affinity of hemoglobin for oxygen. In other words, it increases the release of what? Of oxygen from hemoglobin. So it helps in the release of oxygen from hemoglobin. So what happens that when this is produced, 2,3 biphosphoglycerate, when it is produced in the red blood cell, it's going to bind to the allostatic site of hemoglobin. And when it binds to the allostatic site of the hemoglobin, it's going to tell hemoglobin to offload oxygen. So you find that during strenuous activities in the muscle, because in the muscles there is low level of oxygen during strenuous activities, so the body wants to use that oxygen that you have, the little oxygen that you have. So what happens that we are going to increase the amount of 2,3 biphosphoglycerate. So this molecule, it is increased in the muscle cells when you're doing strenuous activities. Why? Because you have got low level of oxygen. Therefore, that low level of oxygen that we have, we need to offload it. We need to offload it so that it is used. So what will happen that we are going to produce 2,3 biphosphoglycerate so that we increase the rate of offloading that oxygen that is binding to the hemoglobin molecules. So this 2,3 biphosphoglycerate, your takeaway should be, it helps in release of what? Of oxygen from hemoglobin. So high level of 2,3 biphosphoglycerate will increase the release of oxygen from hemoglobin. Okay? So now, some of 2,3 biphosphoglycerate is converted back to 3-phosphoglycerate by an enzyme which we call 2,3 biphosphoglysylate phosphatase. So a phosphatase enzyme, you know what it do? It removes an inorganic phosphate. So we're going to remove a phosphate. That is what it is doing. So 2,3 biphosphoglysylate phosphatase, it's an enzyme that is going to remove that phosphate. So when we remove a phosphate, what are we going to form? We are going to form a molecule called 3-phosphoglycerate. So the Rapaport Leubering cycle, this is simply a shunt. So it is a shunt. So it starts from here, from 1,3 bisphosphoglycerate, we form 2,3 biphosphoglycerate, then you get back to 3-phosphoglycerate. Okay? It's a short pathway. It's a very short cycle. Now, we've seen that 1,3 base phosphoglycerate, it can either enter the Rapaport Leubering cycle or it can proceed in glycolysis to form a molecule that we are calling 3-phosphoglycerate. So this is an enzyme that is called phosphoglycerate kinase. So this enzyme is important. And whenever you see an enzyme ending with kinase, you should know that that enzyme it is either involved in the removal or addition of a phosphate. Okay, so that enzyme, a kinase enzyme, it is involved in the removal or addition of a phosphate. So what happens here that this phosphoglycerate kinase is going to remove a phosphate from where? It's going to remove a phosphate from 1,3 bis phosphoglycerate. So which phosphate are we removing? We're removing the phosphate that is at carbon number 1. So that phosphate there at carbon number 1, we are going to remove it. And when we're taking it, we are going to add it to ADP so that we form ATP. Okay, so here we are forming ATP at that stage. Okay, that is our first ATP to be produced. So we are forming ATP at that stage or at that step. Now, when you form 3-phosphoglycerate, what is happening, guys? There's another enzyme here which is called phosphoglycerate mutase or phosphoglycerol mutase. So what is this enzyme doing? What is the mutase doing? Remember from the Rapaport Leubering Pathway, we said that mutase enzymes, they simply shift the phosphate group or the phosphate. So it's going to shift the phosphate from where? From carbon number 3 to carbon number 2. That's what it's doing. So now what we call this molecule? This molecule we call it 2-phosphoglycerate. So you can look at that diagram that we have there. So you can look at that illustration that we have there. You can see that the phosphate is at carbon number 3, here phosphate is at carbon number 2. So we've shifted the phosphate from carbon number 3 to carbon number 2. That's what is happening here. Okay, so this is called 2-phosphoglycerate. Now, from 2-phosphoglycerate, we can form what we call phosphoenol pyruvate by an enzyme that is called enolase. Then from here, from phosphoenol pyruvate, we are going to form pyruvate. There's an enzyme that is called pyruvate kinase. Now, what is this enzyme doing? You can look at the phosphate there. Where have they gone? Where have these phosphates gone? They have been removed. Now, where have they gone? They're going to go to ADP so that we form ATP. So even here, we are forming ATP. Okay? We are forming ATP from ADP. So pyruvate kinase, this is an important enzyme. Now, what regulates that enzyme? You guys, you should know regulatory steps. The regulatory steps of this pathway, which is the glycolysis pathway, we have got step 1. So step 1 is very important. So step 1, where we've got hexokinase. That is an important step. What else? Step 3, the one I talked about a lot. So step 3, that's why we have got phosphofructokinase 1, PFK1. That is step 3. That is another regulatory step. Very important. What's the other step? This step, where I've got pyruvate kinase. So you can forget anything else, but don't forget this step, don't forget third step, and don't forget step. And you guys, you should pay particular attention to arrows. Just that the arrows here, they're not perfect, but you guys, you should know that this part here, it is reversible. Okay, that is what you should know. You should know that this part here, it is reversible. That part, it is reversible. That part, it is reversible. That step is reversible. But the last step is not reversible. The third step is not reversible. And the first step is not reversible. What is this implying? It's simply implying that these are regulatory steps. Okay? For us to make glucose from glucose 6-phosphate, we need to move a different path, a different route. For example, the one that is used in glycogenesis. You can't directly convert glucose 6-phosphate to glucose. So hexokinase, it is a regulatory step. Phosphofructokinase or PFK1 enzyme, it is also part of the second regulatory step. Okay, so the third step, this is another regulatory step. The other regulatory step I talked about, I mentioned, is what? Is the last step, where we have got pyruvate kinase enzyme. Now, what is regulating pyruvate kinase enzyme? What activates it and what inhibits it? So the inhibitor of this enzyme, we have got ATP. ATP will inhibit that enzyme. What else? R9. R9, it is an inhibitor of that enzyme as well. Now, what activates that enzyme? The activator of that enzyme is fructose 1,6 bisphosphate. That is the activator of that enzyme. So when you talk about regulatory steps, you guys should know that they have activators or regulators. In other words, they have regulators. Now, what are those regulators? So for example, pyruvate kinase, the activator is fructose 1,6 bisphosphate, the same molecule that was produced in the third step. That is the activator. The inhibitor is ATP and alanine. When you've got a lot of ATP, it means that you don't need glycolysis. Why should it continue? So it's going to be inhibited. Okay? Now, how many ATP are we producing from this pathway? That's the other question. How many ATP are we producing? So when you're calculating the amount of ATP, we need to know that at the beginning, we use 2 ATP in the first step and the third step. So when you look at the first step here, we used 1 ATP. In the third step, we use another ATP. So during the first 3 steps, we used what? We used 2 ATP. Okay? So 2 ATP were used. Now, how many ATP are we making? The direct ATP that we are making, if you look at this here, there's an enzyme that is called phosphoglycerate kinase. This enzyme, it said it's making ATP. And remember what I said? I said everything doubles. So we are making 2 ATP there. Where else are we making ATP? We are making 2 more ATP molecules here because we're saying that everything doubles. So even here, we are making 2 more ATP molecules. Okay, so there we're making 2 ATP molecules and there 2 ATP molecules. How many direct ATPs are we making in total? We are making 4 ATP. Okay, we are making 4 ATP. Now, what is our profit? If we're using 2 ATP at the beginning and we're making 4 ATP at the end, what is our profit? What is our benefit? Okay, so our profit is 4 minus 2. So the net amount of ATP made is 4 minus 2, the one that we used at the beginning. So the net amount of ATP produced is what? Is 2 ATP. 2 ATP. That's the net of direct ATP produced. Direct ATP, okay? Mark that word, direct. Now, remember we are also making 2 NADH. So these 2 NADH molecules, they can be taken to the electron transport chain, where they can be converted into what? Into ATP. So directly, we are making 2 ATP. That's the net, okay? So when they ask you what's the total of ATP produced from glycolysis, what's the total of direct ATP produced from glycolysis? It is 4 ATP. What is the net of direct ATP produced from glycolysis is 2 ATP. So the difference between total and net. Okay? So when you say total, it's where we just everything that we are producing without thinking or subtracting this that we use at the beginning. Okay? So without subtracting what we use at the beginning, without thinking of this, we just add everything we produced and forget. That is a total. Now, when we remove what we used at the beginning, we remove that too. What we are coming up with or what we are remaining with, that is our net. So the net of direct ATP produced, the net amount of direct ATP produced is what? Is 2 ATP. The total is 4 ATP. Now, let's also add the indirect ATP produced. Okay? How many ATP are we producing indirectly through these 2 NADH molecules? So we should know, guys, that 1 NADH is equal to 2.5 ATP. So if we are making 2 NADH, it is the same as what? It's the same as 5 ATP because 1 NADH is equal to or equivalent to 2.5 ATP. In some books, you're going to see they have put 3, but according to the current researches, they found out that 1 NADH is equal to 2.5 ATP and not 3. So when you find 3 on Google or in books, don't be confused. You should stick to 2.5. 1 NADH is equal to 2.5 ATP. Now we are making 2 NADH, so that is equivalent to what? That is 2 times 2.5 ATP. Therefore, 2 NADH is equivalent to 5 ATP. Okay, so 2 NADH that we are producing in glycolysis is equivalent to what? Is equivalent to 5 ATP. So you guys, you should forget about pyruvate because these 2 pyruvate molecules that are produced from glycolysis, they are used in TCA cycle. So when you're talking about glycolysis only, and just to calculate the amount of ATP produced, you should just consider NADH and the direct ATP that was produced, the one that we talked about here. Okay, so now from NADH, we form 5 ATP indirectly. Okay? And then we formed 2 ATP, that's the net amount of direct ATP. So 2 ATP plus 5 ATP, what are we getting? So 2 ATP plus 5 ATP. How many are we getting? We are getting 7 ATP. So 7 ATP, that is our net amount of ATP produced in glycolysis. So 7 ATP, that is the net amount of ATP produced in glycolysis, including that that is coming from NADH indirectly. So guys, this was an amazing lecture. Hope you've gotten things from this lecture. And make sure that you subscribe to my YouTube channel. Make sure that you share this video, comment, tell me what you think about this video, and I would really appreciate. See you next time.